Hello students, today I am going to cover the 11th ncert of chapter 2nd, which is biological classification. Let's begin. Chapter Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also he also divided animal into two groups, those which had red blood and those that did not. The Linnaeus time, a two-kingdom system of classification with Plantae and Mammalia kingdom was developed that include all plant and animal respectively. This system did not distinguish between eukaryote and prokaryote, unicellular, multicellular organism and photosynthetic green algae and non-photosynthetic fungi organism. Classification of organism into plant and animal was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Beside gross morphology, a need was also felt for including other characteristics like cells, structure, natures of wall, mode of nutrition, habitat, method of reproduction, evolutionary relationship, etc. Classification system for living organisms have hence undergone several changes over the time. Though plant and animal kingdom have been a constant under all different systems, the understanding of what group organism be included under this kingdom have been changing. The number and natures of other kingdom have also been understood differently by different scientists over the time. Characteristic of five kingdom: character, cell type, Monera prokaryote, Protista eukaryotic, fungi eukaryotic, Plantae eukaryotic, Animalia eukaryotic. Cell wall: Monera non-cellulosic polysaccharide amino acid, Protista present in some, fungi present with chitin, Plantae present cellulose, animal absent. Nuclear membrane: Monera absent, Protista present, fungi present, Plantae present, Animalia present. Body organization: Monera cellular, Protista cellular, fungi multicellular or loose tissue, Plantae tissue or organ, Animalia tissue, organ or organ system. Mode of nutrition: Monera autotrophic. Mode of nutrition: autotrophic. Mode of nutrition: autotrophic, chemosynthetic and photosynthetic, and heterotrophic, saprophyte, parasite. Autotrophic, photosynthetic. Protista: autotrophic, photosynthetic and heterotrophic. Fungi: heterotrophic, saprophytic, parasite. Plantae: autotrophic, photosynthetic. Animalia: heterotrophic, holozoic, saprophytic. R. H. Whitaker R. H. Whitaker, 1969, proposed a five-kingdom classification. The kingdoms defined by him were named Monera, Protista, fungi, plantae, animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 give a comparative account of different characteristics of five kingdoms. The three-domain system have also been proposed that divided the kingdom Monera into two domains, leaving the remaining eukaryotic kingdom in the third domain and thereby a six-kingdom classification. You will learn about this system in detail in higher classes. Let us look at this five kingdom classification to understand the issue and consideration that influence the classification system. Earlier classification system include bacteria, blue-green algae, fungi, mosses, fern, gymnosperm and angiosperm under plant. The character that unified this whole kingdom was that all the organisms included have a cell wall in their cell. This placed together group which widely differ in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae cyanobacteria with other groups which were eukaryotic. It also grouped together the unicellular organism and the multicellular one, say for example Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic group plant, though they also show a characteristic difference in their wall composition. The fungi had chitin in their wall while the green plant have a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera and the unicellular eukaryotic organisms were placed in kingdom Protista. The kingdom Protista had brought together Chlamydomonas, chlorella, early placed in algae within plant and both having cell wall, with paramecium and amoeba, which were early placed in animal kingdom which lack cell wall. It has put together organisms which in early classification were placed in different kingdoms. This kingdom of changes will take place in future too, depending on the improvement in our understanding of characteristic and evolutionary relationship. Over time an attempt has been made to evolve a classification system which reflect not only the morphological, physiological and reproductive similarity but is also phylogenetic, that is based on evolutionary relationship. In this chapter, we will study characteristic of kingdom Monera, Protista, fungi of the Whittaker system of classification, the kingdom Plantae and Animalia, commonly referred to as a plant animal kingdom respectively. We will be dealt separately in the chapter 3 and 4. Kingdom Monera. Bacteria are the sole member of kingdom Monera. They are the most abundant microorganism. Bacteria occur almost everywhere. Hundreds of the bacteria are present in handful of the soil. They also live in extreme habitats such as hot spring, desert, snow, deep ocean where very few other life forms can survive. Many of them live in or on other organisms as a parasite. Bacteria are grouped under four categories based on their shape: the spherical coccus, plural cocci, the rod-shaped bacillus, plural bacilli, the comma-shaped vibrium, plural vibrio, and the spiral spirillum, plural spirilla. Figure 2.1. Bacteria of the different shapes. Though the bacteria structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria are as group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, They synthesize their own food for inorganic substance. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of the bacteria are heterotroph, that is, they depend on other organisms or on dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty area halophiles, hot spring thermoacidophile, and marshy area methanogens. Archaebacteria differ from other bacteria in having different cell wall structure, and this feature is responsible for the survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cow, buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. Eubacteria. There are thousands of the different bacteria on or true bacteria. They are characterized by presence of rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to a blue-green algae, have chlorophyll A similar to green plant and are photosynthetic autotroph. Figure 2.2. The cyanobacteria are unicellular, colonial or filamentous, freshwater, marine or terrestrial algae. The colony are generally surrounded by gelatinous sheet. They often form bloom in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in special cell called heterocyst, example nostoc and anabena. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrate, nitrites, and ammonia and use the release energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are handful of many, making curd for milk, production of antibiotic, fixing nitrogenous in leguminous root, etc. Some are pathogen, causing damage to the human being, crop, farm, animal, and pet. Chlorella, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. The bacteria reproduce mainly by the fission. Figure 2.2, Figure 2.3. Sometime under unfavorable conditions, they produce spore. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cell known and can survive without oxygen. Many mycoplasma are pathogenic in animal and plant. Kingdom Protista. All single-cell eukaryotic are all single-cell eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be photosynthetic protistans to one biologist may be a plant to the other. In this book, we include chrysophyte, dinoflagellate, euglenoid, slime mound, and protozoan under Protista. Members of the Protista are primarily aquatic. This kingdom form a link with other dealing with plant, animal, and fungi. Being eukaryote, the protista cell body consists of well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protista reproduce sexually and asexually by a process involving cell fusion, zygote formation. Chrysophyte. This group includes diatom golden algae decimate. They are found in freshwater as well as marine environment. They are microscopic and float passively in water current, that is plankton. Most of them are photosynthetic. In diatoms, the cell wall form two thin overlapping shells which fit together as a soap box. The walls are embedded with silica and thus the wall are indestructible. Thus, diatoms have left behind large amount of cell wall deposition in the habitat. This accumulation over billions of the year referred to as diatomaceous earth. Being glittery, this soil is used in polishing, filtration of oil and syrup. Diatoms are the chief producer in ocean. Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, red depending on the main pigment present in their cell. The cell wall have stiff cellulosic plate on the outer surface. Most of them have two flagella, one longitudinally and other transversely in a furrow between the wall plate. Very often, red dinoflagellate, example glanulax, under such rapid multiplication that they make the sea appear red. Red tide toxins released by such large number may even kill other marine animals such as fishes. Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of cell wall, they have protein-rich layer called pellicle which make their body flexible. They have two flagella, one short and one long. Though they are photosynthetic in the presence of sunlight, when deprived of the sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigment of glenoid are identical to those present in higher plants. Example, Euglena, figure 2.4b. Slime mold. Slime mold are saprophytic protists. The body moves along decaying twigs and leaves engulfing organic material. Under suitable conditions, they form an aggregate called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiate and form fruiting body bearing spore at their tip. The spore possess true walled. They are extremely resistant and survive for many years. Even under adverse conditions, the spores are dispersed by air current. Protozoan. All protozoans are heterotroph and live as predator or parasite. They are believed to be primitive relative of animal. There are four major group of protozoan. First, amoeboid protozoan. These organisms live in freshwater, seawater or moist soil. They move and capture their prey by putting out pseudopodia, false feet. As amoeba, marine form have silica cell on their surface. Some of them, such as ant amoeba, are parasite flagellated protozoan. The members of this group are either free-living or parasitic. They have flagella. The parasitic forms cause disease such as sleeping sickness. Example, trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms because of the presence of hundreds of the cilia. They have a cavity gullet that open to the outside of the cell surface. The coordinate movement of row of cilia causes the water laden with food to be steered into gullet. Example, Paramecium, figure 2.4d. Sporozoan. These include diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malaria parasite, which cause malaria, a disease which has staggering effect on human population. Kingdom fungi. The fungi consist a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread, rotten fruit. The common mushroom you eat and toadstool are also fungi. White spots seen on mustard leaf are due to the parasitic fungus. Some unicellular flagella, example yeast, are used to make bread, beer. Other fungi cause disease in white rust causing puccinia is an important example. Some are the source of antibiotic example. Penicillium fungi are cosmopolitan and occur in air. Some are the source of antibiotic example. Penicillium fungi are cosmopolitan and occur in air, water, soil and on animal and plant. They refer to grow in warm and humid place. Have you ever wondered why we keep food in refrigerator? Yes, it is to prevent food from growing bad due to bacterial or fungal infection. With the exception of yeast, which are unicellular, fungi are filamentous. Their body consists of long, slender, thread-like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tube filled with multinucleated cytoplasm. These are called syncytic hyphae. Others have septa or cross wall in their hyphae. The cell wall of the fungi are composed of chitin and polysaccharide. Most fungi are heterotrophic and absorb soluble organic matter from dead substance and hence are called saprophyte. Those that depend on living plant and animals are called parasite. They can also live as symbiont in association with algae as lichen and white and with root of higher plant as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, budding. Asexual reproduction is by spores called conidia or sporangiospore or zoospore, and sexual reproduction is by oospores, ascospore and basidiospores. The various spores are produced in distinct structure called fruiting body. The sexual cycle involves the following three steps. First, fusion of protoplasm between two motile and non-motile gamete called plasmogamy. Second, fusion of two nuclei called cryogamy. Third, meiosis in zygote result in haploid spores. When a fungus reproduces sexually, two haploid hyphae of compatible mating type come together and fuse. In some fungi, the fusion of two haploid cells immediately result in diploid cell, that is 2N. However, in other fungi, Ascomycetes, Basidiomycetes, an intervening dikaryotic stage N plus N, that is 2 nuclei per cell, occurs. Such a condition is called a dikaryon and the phase is called dikaryon phase of the fungus. Later the parental nuclei fuse and the cell become diploid. The fungi form fruiting body in which reduction division occur lead to the formation of the haploid spore. The morphology of the mycelium, mode of spore formation and fruiting body form the basis for division of kingdom into various classes. Phycomycetes. Members of Phycomycetes are found in aquatic habitat and on decaying wood in moist and damp place or as obligate parasite on the plant. The mycelium is aseptate and cynocytic. Asexual reproduction takes place by zoospore, that is motile, or by aplanospore, that is non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous, or dissimilar, anisogamous or oogamous. Some common example of mucor, figure 2.5a, rhizopus, the bread mold mentioned earlier, and the albugo, the parasitic fungi, and the mustard. As Ascomycetes, commonly known as sac fungi, the Ascomycetes are mostly multicellular, example Penicillium, or rarely unicellular, example yeast, Saccharomyces. They are sporophytic, decomposer, parasitic, or coprophilous, growing on dung. Mycelium is branched and septate. The asexual spores are conidia produce exogenously on the special mycelium called conidiophore. Conidia on germination produce mycelium. Sexual spores are called ascospore, which are produced endogenously in sac-like asci single ascus. These asci are arranged in different type of fruiting body called ascocarp. Some examples are Aspergillus figure 2.5b, Calviceps, and Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered delicious. Basidiomycetes, commonly known for of Basidiomycetes, are mushroom, bracket fungi or puffball. They grow in soil, on log and tree stump and in living plant body as parasite. Example rust and smut. The mycelium is branched and septate. The asexual spore are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strain or genotype. The resultant structure is dikaryotic, which ultimately give rise to basidium. Cryogamy and meiosis take place in basidium, producing four basidiospores. The basidiospores are exogenously produced on the basidium pleura basidia. The basidia are arranged in fruiting body called basidiocarp. Some common members are agaricus mushroom figure 2.5c, Eustilago smut, and Puccinia rust fungi. Deuteromycetes, commonly known as imperfect fungi because only the asexual or vegetative phase of these fungi are known. When sexual form of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stages have been given one name and placed under Deuteromycetes and sexual stage another and placed under another classes. Later, when the linkage was established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect sexual stage of member of Deuteromycetes were discovered, they were often moved to Ascomycetes or Basidiomycetes. The Deuteromycetes reproduce only by asexual spore known as conidia. The mycelium is septate and branched. Some members are saprophyte or parasite, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Colatotrichum, and Trichoderma. Kingdom Plantae. Kingdom Plantae include all eukaryotic chlorophyll-containing organisms commonly called plant. A few members are partially heterotrophic, such as insectivorous plant or parasite. Bladderwort and Venus flytrap are examples of insectivorous plant and Cuscuta is a parasite. The plant cell have eukaryotic structure with prominent chloroplast and cell wall mainly made up of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae include algae, bryophyte, pteridophyte, gymnosperm, and angiosperm. Life cycle of the plant have two distinct phases: the diploid sporophytic and haploid gametophytic that alternate with each other. The length of haploid and diploid phases and whether these phases are free living or dependent on other vary among different groups in plant. This phenomenon is called alternation of generation. You will study further detail of this kingdom in chapter 3. Kingdom Animalia. Kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cell lack cell wall. They directly or indirectly depend on the plant for food. They digest their food in an internal cavity and store food reserve as glycogen or fat. Their mode of nutrition is holozoic by ingestion of the food. They follow a definite growth pattern and grow into adults that have a definite shape and size, have higher form, show elaborate sensory and neuromotor mechanisms. Most, most of them are capable of locomotion. The sexual reproduction is by copulation of male and female, followed by embryological development. Silent features of various phyla are described in chapter 4. Virus, viroid, prions, and lichens. In the five kingdom classification of the Whitaker, there is no mention of lichen and some cellular organisms like virus, viroid, and pyrons. These are briefly introduced here. All of us who have suffered the ill effect of the common cold or the flu know what effect virus can have on us. Even if we do not associate it with our condition, virus did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having a inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? Virus means venom or poisonous fluid. Dmitry Ivanovsky, 1892, recognized certain microbes as causal organisms of mosaic disease of tobacco, figure 2.6a. These were found to be smaller than bacteria because they passes through bacteria-proof filter. M.W. Bijonic, 1898, demonstrate that the extract of the infected plant of tobacco could cause infection in healthy plant and named the new pathogen virus and called the fluid as Contagium vivum fluidium, infectious living fluid. M.W. Stanley in 1935 showed that virus could be crystalline and crystal consist largely of protein. They are inert outside the specific host cell. The virus are obligate parasite. In addition to the protein, virus also contain genetic material that could be either RNA or DNA. No virus contain both RNA or DNA. A virus is nucleoprotein and the genetic material is infectious. In general, viruses that infect plant have single stranded RNA and viruses that infect animal have either single or double stranded RNA or double stranded DNA. Bacteria, viruses or bacteriophage. Virus that infect the bacteria are usually double-stranded DNA virus. Figure 2.6b. The protein coat called capsid made up of small subunit called capsomere protect the nucleic acid. The capsomere are arranged in helical or polyhedral geometric form. Virus cause disease like mumps, smallpox, herpes, and influenza. The aid in human is also caused by virus. In plant, the symptoms can be mosaic formation, leaf curling, yellowing, and vein clearing, dwarfing, and stunted growth. Viroid. In 1971, T. O'Diner discovered a new infectious agent that was smaller than virus and caused potato spindle tuber disease. It was found to be free RNA. It lacked the protein code that it found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight. Pyrons in modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consist of abnormal folded protein. The agent was similar in size to viruses. These agents were called pyrons. The most notable diseases caused by pyrons are bovine spongiform encephalopathy (BSE), commonly called mad cow disease in cattle, and its analogous variant Jacob disease (CJD) in human. Lichen are symbiotic association that is mutually useful association between algae and fungi. The algal component is known as phycobiont and the fungal component as mycobiont, which are autotrophic and heterotrophic respectively. Algal Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrient and water from its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichen are very good pollution indicator. They do not grow in polluted area. Summary: biological classification of the plant and animal was first proposed by Aristotle on the basis of simple morphological character. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whitaker proposed an elaborate five-kingdom classification, Monera, Protista, Fungi, Plantae, Animalia. The main the main criteria of the five kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationship. In the five kingdom classification, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista include all single-cell eukaryotes such as chrysophyte, dinoflagellate, euglenoid, slime mold, protozoan. Protists have defined nucleus and other member-bound organelle. They reproduce both asexually and sexually. Members of kingdom fungi show a great diversity in structure and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes are the four classes under this kingdom. The Plantae include all eukaryotic chlorophyll-containing organisms. Algae, bryophyte, pteridophyte, gymnosperm, and geosperm are included in this group. The life cycle of the plant exhibits alternation of generation, gametophytic and sporophytic generation. The heterotrophic eukaryotic multicellular organisms lacking a cell wall are included in the kingdom Animalia. Students, if you want the notes of this chapter, then my Telegram channel you will get DPP also of this chapter and PYQ question of this chapter of 20 year and short note also.